A warm welcome to all of you to coming to this lunch lecture, which is a part of the International Open Access Week. My name is Niclas Jareborg and I'm the library director of the University Library. And it's my great pleasure to open this event and introduce our speaker, Dr. Niclas Jareborg. He comes from the National Bioinformatics Infrastructure Sweden, NBIS, and will talk about open access and data management in the life sciences, which is a very, very interesting topic and challenging perhaps. I think that we will hear about the opportunities and more about the challenges that are faced with the life sciences when large data sets should be open for use for other scientists. The stage is yours.
Thank you.
Warm welcome.
And thank you for the opportunity to come here and talk about this. I'll start off with talking a bit more about where I'm from and the organization I belong to. So we're part of the SciLifeLab, and I assume all of you know what SciLifeLab is, but I'm going to anyway say that it's about now around 1,200 staff associated with the SciLifeLab, and it's hosted by 3 Stockholm universities and Uppsala University, and have sites here just beside the KI campus and in Uppsala University. There are also affiliated sites at other Swedish universities, and this will expand more in the coming years, I believe. Close this. And the mission is to be a national hub for technology platforms for life sciences, to provide cutting-edge infrastructure for life science researchers. Also to be a strong collaborative research community that can use those platforms. And thirdly, to impact society at large where it can. It's headed by Olli Kallioniemi and Lena Claesson-Welch. And Olli started 2 years ago, basically. Before that, it was Mattias Ullén at the KTH that was one of the initiators of the center. Our activities belong to this national service, that the Swiss Army knife for Swedish life science researchers. And it's made up of a number of infrastructure platforms. One of those is the bioinformatics infrastructure, which was formed last year from a couple of different other smaller bioinformatics infrastructures. The bioinformatics sort of goes across all the different platforms. We're not going to go into what you can do, but the message is there are lots of technologies, and it's open for everyone to use those technologies. That's basically it. However, the coordination of how you as a researcher access those facilities has not been very clear and maybe not very easy to coordinate. So the user has to coordinate that him or herself. But it was this year we've formed something called the Data Office that will aim to coordinate better between the platforms and guide the researchers in how they can use these platforms better. But also work at making the data that these platforms produce more visible. So they will provide data coordination. They will try to track the data that's been produced by the platforms for Swedish researchers, help with access to data if possible, and also give support when it comes to where data should be deposited. But so that's SciLifeLab. Getting more specific, our organization, I think we've made a movie, so I got to show the movie. nice, isn't it? So we have one national infrastructure. So we are the SciLifeLab bioinformatics platform, but we're also spread over all major universities in Sweden. And we're financed by those universities. We're also financed by SciLifeLab directly, Knut and Alice Wallenberg-Stiftelsen, the Research Council, and also to some extent by user fees. And the user base is the Swedish university academic users, also hospitals and companies. And we interact internationally. And we do support, or our activities are basically three legs. Support, analysis support for researchers, tools for the researchers to help their analysis, and also provide training on how to use these. Data management, which is my task, goes across all of these. Unfortunately, we're not very strong in manpower. It's basically me and part-time. But we collaborate very, very closely with the SciLifeLab data office around these issues. The support, the main support is early in the process to study design. You can have a consultation. Then we have paid support for 800 crowns per hour. Then there's also a long-term track, which is a peer-reviewed. You apply 3 times a year, and if you get granted that, you get free support for 500 hours and an expert bioinformatician in your research project. But that's, yeah, it's not easy to get those. We have also some part of our personnel that works very closely to the SNCC computing organization around computing and storage. I should also mention that we are also heavily involved in international collaborations, the main then being Elixir, which is the European bioinformatics infrastructure network. It's made up of over 20 members, and try to set up a data infrastructure for the life sciences in Europe. And it has a number of platforms. And the idea is that each node from each country contributes with the resources it has, and we work together across the whole infrastructure to get a common set of data resources and ways to access the tools. Yes, let's go. So here we are. Now I've thrown a lot of organizations in your face. And this is a bit of a problem because it's very messy. This is the reality of the support infrastructure landscape, basically. We're a part of everything. And one complication here is that in this picture, I think it's only the Swedish universities that are legal entities, which makes it hard to do things. But this is what we live with. Enough of organization. Over to the life sciences. As you are well aware, the life sciences are very technology heavy. And in recent years, there have been fantastic progress and developments in this. And the big players are, not surprisingly, DNA sequencing, imaging, proteomics. And if we take DNA sequencing, the first human genome took 13 years to complete, cost about $1 billion, depending on how you calculate it. Nowadays, we're down at the $1,000 cost for sequencing a human genome. And it's done in 1 or 2 days. This, of course, creates some problems. It's now very easy to generate more data than you can handle. And DNA sequencing is projected to overtake all other research areas. Even, I mean, in 10 years it's going to be more genomic data than the YouTube or astronomical data and other physics data. It's growing very rapidly, and it's a complicated beast. It's not straightforward to store, it's not to distribute, to analyze. And we are close to some sort of tipping point where it's often more costly to analyze and manage and store the data than to generate it. And this is, I think, the problem we got now is that not everyone is aware of this when they order the data from the sequencing facility, for example. You have money, you spend all your money on getting the samples and getting the DNA sequences run. But you don't budget for what you need next. So, data management in the life sciences. I think it's useful to talk about this structured around the data life cycle. And you can draw this in many different ways. I hope you can see this, but you start by planning what you're going to do. You generate your data. You study and analyze it. You store it sort of midterm and share it with colleagues maybe. You put it away when you're done there for longer archiving. And hopefully you also make it public for others to reuse. Now, this is of course a bit contrived. It's seldom this exactly these blend into each other and so on. But I think it's useful to look at things in this way. So, at the planning and design stage, what should you do when it comes to data management? Well, it's a good thing to look at what type of data will I be producing, for example. Where should I store that? Also, thinking about how can I describe the data I will generate? What are the samples, how the experiments are going to be done, and for that to use standards. This will probably become a lot more formalized in the coming years when so-called data management plans will become more and more common. A data management plan is putting down in some form what would I collect, how will I organize it, how will I document it, where will I store it and preserve it, how will I share it with others, under what conditions can I share it, what will it cost. And this is, doing one of these plans is less getting the right answers, it's more of sort of an exercise of finding out what you don't know. And there is a stick here. Funders will start demanding it when you apply for research funds. But that you should maybe, if you can have the mindset that this is something that is beneficial for my own research, to actually beforehand think through that I need somewhere to store it. I need to know what types of data I'm going to be generating and how I'm going to describe it. In the European collaboration, we are working on a pilot around a data management tool that can help us. So this is sort of the idea is have a decision tree. You get questions that you sort of select choice answers from, and that will give you guidance on how to handle certain questions. This is just in its infancy. There are talks about generating a Swedish resource, a national resource for data management plans, but that's maybe so far just thoughts in some people's heads, I think. The US and the UK in particular are very far ahead in this, actually. We're a bit behind here. And I'd say our neighbors, at least, especially Finland, is very well ahead of us in this area. That was planning. Then you generate your data and you get it, so you're going to analyze it. How can I think about managing my data at that stage? Well, one thing to have in your back of your mind is that if you can organize things in your projects so anyone that is not familiar with it can understand what you've done, that's a good way to look at things. And, well, that might be you yourself in 3 months, because we tend to forget what we have done. And whatever you do, you have to do again and again and again, basically. And if you do this, organize things, it will in most cases actually speed up your research, because you waste less time trying to figure out why you did something when. And this is, of course, also critical to make things reproducible. And you've probably heard of, some consider we have a reproducibility crisis at the moment, when meta-analyses are done in not only life sciences, but in many different areas of research. About half of the results are not possible to repeat. Right. So, how should we organize things? You have your data and you put it in a folder. Great. Excellent. But then you fiddle and tweak it a bit. So but you don't want to throw away the old one. So you keep it around. And you do this some more. And even some more. And you end up in this sort of mess. But let's put some structure to it, so you put it in folders instead, but maybe not. One way, a possible solution, is use dates on your folders. It's one thing. But so that's about your data, but you also need to describe what is the data. And there we have what we call metadata, the data about the data. So how was it generated, from what, experimental conditions, and so on. And in this area, there's something that came up last year, a paper about the FAIR guiding principles for scientific data management and stewardship. So FAIR then stands for findable, accessible, interoperable, and reusable. And not only for humans, but also for machines. And this took off, really. About 6 months later, sort of the bigwigs in the world hooked onto this. And this is, the leaders of our world wants research data to be fair. And the European Commission has initiated something they call the European Open Science Cloud, which by some is said not to be only European, not only open, not only science, and it's not a cloud. But anyway, the idea is to have enable trusted access to services, systems, and reuse of shared scientific data across disciplinary, social, and geographical borders. Very grand. How are you going to do it? The FAIR bits here are a cornerstone of this, and there was this declaration released by the European Commission around this. So this will guide a lot of how research funding will be what's expected to get research funding is that you adhere to this and play into this sort of ecosystem that will hopefully evolve in the coming years. So how are you going to be fair then? So these are quite high-level descriptions for the different aspects here, but a large number of them mention the metadata aspect as being very important. So for that, so how do you describe things? It's very important, or it makes things so much more useful if you can use standards. For example, in the forms of controlled vocabularies in the ontologies. Does anyone know what a controlled vocabulary is? Librarians, or an ontology. So a controlled vocabulary is a set of terms where the term describes some sort of phenomenon. And the idea is that you have one term to describe the phenomena, not almost the same, or slightly different. A former head of the European Bioinformatics Institute said, it's probably 20 years ago, a biologist would rather share a toothbrush with another biologist than share a genome, which has created quite a lot of problems in life science, I'd say. An ontology then is putting a structure on this list, sort of how the terms relate to each other. We don't have to go into the details. But so in life sciences, people have thought about, oh yeah, we got to fix this. But there must be some way of having a universal one, so we introduce yet another one. So currently there are over 600 content standards in the life sciences. About 350 of them are sort of terminologies. And how are you, as poor human beings, going to navigate this? It isn't straightforward. There's this site called fairsharing.org. It started as biosharing.org, but they are now expanding. It started with the life sciences, but they're now expanding. So where you can go in and look for standards of terminology standards, but also different databases and policies. And policies might be which standards a database suggests you should use to describe your data, or particular journals. This is, of course, a work in progress, a never-ending thing, but I think it's a useful resource that you can make some parts of your research life simpler. Right, so let's say we now have documented the data we have perfectly. We still need to log what we actually then do with the data. So write notebooks, because you yourself will have to understand what you've done. Others should understand what you've done. So put things in with date stamps. If you're doing computation, write down what was the computation you did, so on. And where shall I do this? Well, of course, you can use good old paper notebooks, word processor files, text files. There are electronic lab notebooks sold by companies for a lot of money. What quite recently popped up what you could call interactive electronic notebooks, where you can mix text with code. There are some, the big ones are Jupyter, and another is called R notebooks. What you do is you write plain text files, and then there's an interpreter on that, so that can run the code you put in the document and have the output automatically appear in the output that you can then format as you wish. And this makes it easy to, if you want to I need to clean up the data. There was some point there that I have to take away. You remove that point, and then the figure for the paper is updated, basically without you having to go through it all again. Right, so we have all these components. How do I structure things? There are people that thought about this. This is a quite nice little paper from almost 10 years ago. But you have your data separate from everything else, your raw data separate from everything else. You have the code that does the work in separate folders. You have put out the outputs and results in your notebooks, also separate from everything else. And document what goes where and why. And I think the important part here is to try to think through where am I going to put these bits and pieces and try to stick to it. There's no perfect setup. There are lots of templates out there. There are some links there you can try. But pick one. Decide with your colleagues how you're going to work with that. Be sure to document, and hopefully you will understand what you've done and will not have to explain for everyone. Right. I assume that many of us here work with human data, which brings me into a bit talking about legislation. When it comes to personal data, there are two laws. One is the Personal Data Act, Personuppgiftslagen, or PUL. All kinds of information that is directly or indirectly referable to a natural person who is alive is personal data. Some of that is classified as being sensitive. And it's prohibited to work on sensitive data. Sensitive data is sort of political opinion, religious, philosophical conviction, etc., as well as data relating to health. But you can handle data for sensitive data for research, so that's sort of okay, good, we can do that. I should mention then that in May, we will have new legislation around this. It's an EU-wide legislation called the General Data Protection Regulation, or GDPR for short. And as a consequence of that, we have several new Swedish laws. The overarching law would be the Data Protection Act, Dataskyddslag, and a specific Research Data Act for research data. And this will then replace the old PUL. Differences are maybe not earth-shattering, but for research, I'd say if we follow the current legislation properly, there shouldn't be any problems. I'm not going to go into what's new in the GDPR. For one thing, I don't grasp it all, and the other is it would take too long time. But one consequence is, with the new legislation, even a university can be fined for not finding the rules. So either 10 or 20 million crowns for not handling personal data properly. Then we have another law about ethical review. So if we're going to do research on humans, we need ethical vetting. The research has to be reviewed, and an informed consent from the research subject. They must understand the risks and consequences of being part of the research and consent to that. This hopefully not anything that's new to you. So why am I talking about this? Well, the big thing is, of course, genetic information. Genetic information from a human is sensitive data. As under the current legislation, it relates to health, and in the new legislation, it's explicitly defining as being sensitive data. And there's no way around it. The genetic identity is an identity that has to be protected, even if it's extremely hard to couple it to the person or the personal number or a name. And this is true even if you think you can anonymize it or pseudonymize it. You have to treat it as sensitive personal data. And it doesn't matter if it's whole genome sequence or parts of exomes or transcriptomes or so. And it's been shown that it's theoretically possible to identify an individual person just from the sequence itself, in some cases. But okay, what are you as a researcher supposed to do? How are you going to handle this properly? The law says the controller, that is the data owner, is liable to implement technical and organizational measures to protect the personal data. The measures shall attain a suitable level of security. But what is that? It's what the data inspection says when they come and look at how you've done it, which doesn't help you in any way. We have taken a stab at this, trying to provide a computing environment at least. So the Swedish National Infrastructure for Computing, SNIC, have launched a system that they call Bianca for doing computation on molecular biology data that opened earlier this year. It was built on a pilot system that my organization set up a couple of years ago that we call Mosler, that's still around. But it's a compute system where we've taken measures to make it up to the security. It's harder to break in, or close to impossible. Nothing is impossible, but it's very hard to hack your way into the system. And when you're in it, you cannot get at any other project's information. So we believe this is a suitable level of security. So those systems are available if you should need them. I would also like to mention an ongoing project run by the Elixir nodes in the Nordics, Iceland not included, under the umbrella of the Nordic e-infrastructure collaboration project. And this is about making it easier for Nordic researchers to collaborate on sensitive data, pooling data from different countries in secure environments. I'm not going to go in. It has technical development parts, interoperability, and also looking at legal bits. I'd like to bring up the use cases. So if you have, as a researcher, want to collaborate with other Nordic researchers, we have support for setting up those collaborations and providing help with the infrastructure and so on. So you can apply for that. These slides will be available later on, I believe, I hope. I will make them. And if not, come contact me and I can help you if you need. Right. Then at the end, if you're a good citizen, you should publish your data. And this is a cornerstone of the open access principles, I'd say, because you want the data to be available long time, and you should be able to refer to the data over long periods of time. So you need some sort of persistent identifiers. You probably heard of the DOIs. If you're looking for journal articles, it's an identifier that will always find the article. You can use DOIs for research data sets as well. And you should make the data discoverable. Somehow make the metadata available. This is okay. Interesting. I'm missing a slide. All right, I'm going to do this. In the life sciences, there is a long tradition of data sharing. even in the, the DNA sequence databases and the protein structure databases were well, PDB, the protein structure database started in '69, and the GenBank and the EMBL database in the '80s. And in the '80s, it became mandatory for most journal publishing that you need to submit your sequencing data. So there's this long tradition. In the Human Genome Project, they sort of went even further, that all the collaborators in that project agreed to automatically release the data immediately, basically, within 24 hours. To publish everything immediately and make it freely available. But in reality, data disappears as it is. There are several studies having looked at how you can get at data, and it drops up pretty rapidly. Either the link that is in the paper leads to nowhere, or the link leads to a version of the dataset which is not the same that was in the paper. So, and this is part of the reproducibility crisis. A large part of the studies that are not reproducible because you cannot find the data. And we have then now, from the European infrastructure, Elixir, made a statement of that we also think this is extremely important to openly share data, and it's a crucial part of good scientific practice. So this is how we will operate and push for that, how things should be done. And the best way, as it's written here, that biological research data should be submitted to the recommended community deposition databases. So the easiest way to make your data fair, findable, accessible, interoperable, and reusable is to use international public repositories, of which there are many. For one thing, that they provide the domain-specific metadata standards. However, even though we have this long tradition of submitting, there was a study, this is 2 years ago, I think, looking at research funded by NIH, and they went through a large number of papers that came out of that research and found that only 12% of those articles mentioned data deposited in international repositories. And they estimate over 200,000 invisible data sets per year being generated. Invisible in the sense that the only way of finding the data is if you find the paper where it's linked to. We did our own survey a couple of years ago with a much smaller set, but users of the National Sequencing Facility. And more than 25% had never submitted their data. And the reasons for this is basically they need help submitting, and it's hard for them to find the right repository. So that's sort of guiding what I do in everyday life to help people find where to put the data. The Elixir infrastructure has also defined a core of data resources that they want to help provide long-term support for, and this is a list here, basically, but they also specified a list of deposition databases that can help you see where you should put your data. And if you have questions, you can always come to us and ask for support. So, but how about sensitive data? Because that shouldn't, we have genetic data from humans, that shouldn't be open. Well, there is this thing called EGA, stands for European Genome Phenome Archive. It's a repository where you can submit your data, but only release it to people that have a legitimate access right to it. If they have ethical consents that are in line with the consents of the original research. So it has a mechanism of asking the data owner if I, as a researcher, can have access to a certain data set. So the metadata about the studies and the samples are findable, but the actual data you have to request and go through a vetting from the data owner. The Elixir infrastructure is also working quite hard on especially human data in many different forms of making it easier to interoperate about that. One part is that we're setting up local instances of the EGA database. So not everything will reside in a central store as it is now, but we will establish local databases where we will host the data. So we will host the Swedish data here. And that will fix some legal problems, actually. But the metadata about the data will be stored centrally and will be searchable centrally. So you can find about studies regardless of where the data is stored. Another way of sharing human data is a resource we set up, initiated by what is called the SweGen project. So that's a reference sequence project for 1,000 human Swedes. It's a representative set of the Swedish genetic variation. So we have this site called SweFrec, where you can get a lot of information, but that's about frequencies and not the individuals. And we're taking more data sets there. If you have human variation data, we can host that. A part of that, a way of accessing that information is through something called beacons, which is comes out of the Global Alliance for Genomics and Health. It's a way of querying a data set. If you've seen a variant at some certain position in the genome and want to know, has anyone else seen this? You can ask, do you in your data set have a T in this position? And you get a yes/no answer. If you are then a clinician, for example, looking for a rare disease, you can then go further and contact the data owners. And the neat thing is this is built into a network, so you can query the whole network at once and get your information. Right, some data might not fit. And there are repositories in these databases, and there are repositories for these as well. So it will give you will publish the data, you will get persistent identifiers, you will in most cases be able to specify some metadata, but they are usually of more of a general kind, so they're not targeted towards life science, and this of course affects discoverability, and so I shouldn't say that they're not fair, they're less fair. Fair is not a qualitative aspect really, because there are degrees to that. And to my knowledge, there aren't any sort of general repositories for sensitive data yet, but I might be wrong. I listed a couple there, basically. I'm getting near the end. Some of you may paid attention that I skipped over 2 parts here. And currently in Sweden, we don't have very good infrastructure support for this part. We have support for compute and storing the data there. And we can help put the data in international repositories. But the long-term storage down here, and especially the archiving, which is the responsibility of the universities to archive the research data, is not catered for really when it comes to these very large scale data sets. However, SUNET, earlier about a year ago, initiated some sort of initiative together with a couple of very big infrastructures, research infrastructures, to put up a national storage solution for this, in which you can sort of manage your data across the process and make it available for computing if you need that, for archiving and for publication. That's at least how we hope it will be. And so this should cover this bit of it. But I think it's mainly a political issue at the moment before this becomes a reality. That was it. Thank you. I'm happy to take any questions if you have them and so on. And just finally like to acknowledge some people where I've stolen most of this information. So, thank you very much.